Welcome everyone who both in the audience here at Heidelberg, but also those who might be online. We've got a really special presentation today focusing on marine mammal conservation. I'll introduce our speakers in a minute, but before I do that, I'd just like to begin by acknowledging the traditional owners on the land which we're meeting on. And here in Heidelberg, we're on the land of the Wurundjeri country, and I pay my respects to elders past and present, and also to the Aboriginal people and the traditional owners on the land which people are coming from online as well, and to any Aboriginal people who might be joining us. My name's James Todd. I'm the Chief Biodiversity Officer and Arthur Rylah Institute Director, and I'll be chairing this seminar with technical support from Andy and Tess. So today I'm really excited about our two guest speakers. We've got Dr. Barbara Taylor and Dr. Jay Barlow. Both Barb and Jay have a wealth of experience working as government scientists in the US and have had a huge influence in how science is used in policy and decision making in the US as well as globally. Their expertise and the research questions they address have considerable overlap with our work here in Victoria and Australia more broadly. Our first presenter will be Dr. Barbara Taylor. Barb was a leading scientist at the National Oceanic and Atmospheric Association, otherwise known as NOAA, I assume, a Southwest Fisheries Science Centre in La Jolla in California. She's leading the global efforts to save the vaquita, which is a critically endangered porpoise in the Gulf of California, from extinction. She's also an expert in decision analysis, and she led the marine mammal genetics program while at NOAA. Today, Barb is going to share her wealth of knowledge on the human dimension of conservation. We will have time for questions at the end of the presentation, so for those people online, please put those in the chat. And for people in the room, we'll have hands up at the end. So thanks, Barb, for being here, first of all, and welcome, and over to you.
Well, good day, everyone. It's a real privilege to be here, and I'm going to start out by saying that when I was telling my mother, who just turned 100, what I was talking about in Australia, and that it was, changing human behavior, her response was, "You don't know anything about that." And my reply back to her was, well, actually, that's the point of my talk. So I'm going to talk today about changing human behavior, and with the marine mammal conservation. This is sort of a reduced form of a plenary that I just gave in Perth. It's actually not a completely novel concept, but a somewhat novel concept. That in fact, we actually have to change human behavior. So I gave a plenary in New Zealand about 12 years ago, and it was talking about the ingredients that we need to be successful in a conservation recipe. And we had a period of tremendous successes between the 1970s and the 1990s, where commercial whaling was curtailed, high seas drift net fishing was curtailed, and the tuna dolphin fishery was largely curtailed. And the ingredients to that recipe were always about the same. It was NGOs, resource users, implementers, including enforcement, legislators, sometimes the courts, and always natural scientists. And so those problems were dealt with very well, but the biggest conservation problem for marine mammals is actually bycatch, accidental deaths in fisheries. And it was not dealing well with that. And there's also issues of habitat degradation and climate change. And so at that point, I suggested that we needed some new ingredients, and educators, media specialists, economists, and fisheries technicians needed to be added to the mix. So we recently published a paper on how have we done. So the IUCN has a cetacean specialist group that deals with whales, porpoises, and dolphins. And we looked at the proportion of threatened species over time, and in 15% were listed as threatened, which means critically endangered, endangered, or vulnerable in 1991, 19% in 2008, and up to 26% in 2021. So that's not a great success rate. We also looked more deeply at the different habitat types, and we broke it into river, coastal, shelf, and open ocean. And all of the species that live in rivers are either endangered or critically endangered. And over half of the species that live in coastal waters are threatened in one of those categories. And so basically, if as a species you must live near humans, you're facing some very serious risks, and it's mostly bycatch. So a paper just recently came out called Changing Human Behavior to Conserve Biodiversity. And it opens with conservation of biodiversity is above all an exercise in human persuasion. Across the more than 40,000 species currently listed as globally threatened, 98% are threatened exclusively by processes driven by human behavior. Yet conservation remains predominantly a natural science discipline, with the social and behavioral sciences playing a secondary role. So the IUCN Species Survival Commission has a group that specializes in conservation planning, and they have embraced what's called the one plan approach. And this was in response to lots of conservation plans sitting on shelves gathering dust and not much action actually happening in the field. And so they felt there was a need for a really substantial change to develop conservation plan in concert with all of the stakeholders so that there was stakeholder buy-in. And they define stakeholder as any individual or group that may affect or be affected by conservation work. So a really critical step is, who do you invite to the table? There's lots of fishers, if in the case of bycatch, but who is going to come to the table to really help in the long run promote conservation? So in late 2022, I attended a workshop that was called The Human Dimensions of Small Cetacean Conservation. And in that workshop, we concluded that one, incorporating human dimension considerations into small cetacean conservation efforts means careful attention to the role of human behavior in causing and mitigating impacts on the animals and their environment. Two, successful strategies for improved dolphin and porpoise conservation must ultimately include actions that result in changes in human behavior. And three, we must work with and for people to identify shared goals in conserving dolphin and porpoise species while ensuring that human communities prosper. So there were many disciplines that were represented at that meeting, and I'm going to go into the details of those in a moment. But meeting those people, like conservation psychologists, which I didn't even know was a job, really hit home to me on the importance of how they are very skilled in doing the things that we were not doing well. So just to give a few of the field definitions, conservation economics is the use of economics to understand the costs and benefits of sustaining natural ecosystems. Its purpose is to accomplish more widespread and lasting conservation by lowering its costs, revealing its benefits, and fitting it within genuine economic development. Conservation sociology is a field that uses sociological theory and research methods to study the relationship between humans and nature and to improve conservation practices and policies. Conservation psychology is the study of how humans interact with nature and how to encourage people to protect the environment. It's an interdisciplinary field that uses the principles of psychology to understand and solve environmental issues. Community engagement is a little bit trickier because it seems to be both a process and a person or persons. So it's a collaborative process that involves working with groups of people to address issues that affect their well-being. The goal is to achieve sustainable outcomes that improve the community and its members. But then I also found things that said it's a bridge between expertise and knowledge of institutions and communities. In the workshop, it was made very clear by all of the social scientists there that what you needed really was a community conservation leader, a trusted translator who lived in the community and could translate between the agencies, the other stakeholders, the scientists, and the community. And this has been a really key element that has been missing in many of the conservation situations that I've dealt with. So I'm going to give a few examples of different species so that we can sort of see what these human dimensions are in context. So the Yangtze finless porpoise, it's critically endangered. There's about 1,200 individuals. Its major threats are shipping, habitat degradation, and it's only present in China, and the major stakeholder is government. And I should say that each one of the co-authors in this talk were specialists in these different species that I'm going to be talking about. And I asked them to basically fill in this table for me, where I first asked what levels of engagement there were in these different science, social science fields of economics, sociology, education and communication, psychology, and community engagement. And you can see that it's very mixed here for China, with only one low in the green, a couple in medium, and a couple in high. But then I asked them what social science expertise was needed to actually reduce the threat. And you can see that these actually, for China, don't match, that the community engagement was high, but it's actually not needed. And the reason why is because China just in 2020 banned all commercial fishing. So the government can make it happen. And so they didn't actually need community engagement, even though they had a lot of community engagement. And it's of the species I'll talk about today, special in that it's the only one that actually is increasing because it dealt with this problem of bycatch in a very authoritarian and effective way. Humpback Atlantic humpback dolphins are critically endangered with about 3,000 individuals. The major threat is bycatch. It's present in at least 14 countries, and the major stakeholder is fishers. And you can see how much more difficult the human dimension is for a species that spans all of these different governments and cultures and languages. And so there's been a lot of work on this species, but there's a long way to go. So they listed all of the social science engagement as low or medium, but in fact, in some countries, they're doing really well, and in other countries, they haven't even started yet. But they listed the need for those social sciences as to reduce the threat as high, almost universally across all of those different disciplines. Lahill's bottlenose dolphin is listed as vulnerable. There's about 600 individuals. The major threat is bycatch, and it's present in 3 South American countries. And again, the major stakeholder is fishers. The level of engagement is very similar to the last situation, where some areas they've made a lot of progress, but generally the level of engagement across the range of the species is low to medium, but high needs in sociology and community engagement. And for vaquita, it's critically endangered, only about 10 individuals remaining. The major threat is bycatch. It's present only in Mexico, and the major stakeholder, again, is fishers. And again, the level of engagement of social sciences is low to medium, but it's strongly needed in sociology, psychology, and community engagement. And finally, the North Atlantic right whale has been listed as critically endangered just since 2020. There are about 350 individuals. The major threats in about equal proportion is ship strikes and gear entanglement. It's present mainly in 2 countries, and the stakeholders are the fishing industry and the shipping industry. The level of engagement here is actually medium to high and matches pretty well with the needs to reduce the threat. So if we take an overview, just looking at the social science field, the areas where the social science was deemed in high need to be able to reduce the threat, you can see that every single one of these species is really different. And that's, I think, a real lesson about the human dimension, is it's going to be different for every single species in every situation. The little hand with the plant is cases where you had medium social science expertise being engaged, but you needed high engagement. And you can see that the field of community engagement, the only place where it wasn't deemed in high need was China. Every place else it was deemed in high need. And there's a bunch of sad faces where there's very low levels of engagement and high need for engagement. And you can see for vaquita, the species that I've worked with most, it's all sad faces. So this same paper had, I thought, a very interesting caution, and that was that the need to influence people has been a significant aspect of conservation work for several decades. However, the focus has often been on raising awareness, improving knowledge, and changing attitudes. Although these factors may be important in certain contexts, a significant body of work suggests that changes to awareness, knowledge, or attitude seldom leads to change in behavior. Indeed, although influencing these attributes may be necessary, these changes are only the initial steps in bringing about behavioral change. True conservation gains and the mitigation of extinction rates can be realized only when change occurs tangibly in the real world. Therefore, emphasizing awareness, knowledge, attitudes, or other intangible indicators to understand or change behavior can be a distraction. These indicators are easier to influence, but frequently are not directly related to behavioral change itself. As a result, they may create an illusion of progress. So again, I asked my co-authors to list whether they had actually metrics for changes in human behavior. And interestingly, most people responded in terms of the biology of the animals that they work with. So they listed abundance and trends in abundance and mortality rates, with a lower number listing sort of what I would call the direct effects of measuring human behavior: enforcement efficacy, amount of gillnet, and uptake of alternative gear. And I won't spend a lot of time here, but I also sort of color-coded the arrows as to how well they were doing. And you can see that for vaquita, the 2 direct measures get a red arrow, which means bad. And basically, there has been good measures of those things in the past, but I think that the government isn't particularly interested in showing how poorly they're doing, so they're no longer measured. So let me conclude with a couple of general thoughts. We're still important, I think, as conservation biologists, because we really need to provide the best available science in a timely fashion. I really think it's important that we communicate our passion and belief that the threats can be sustainably managed. If we don't have faith to say that the animals can survive and recover, then why should people change their behavior? And finally, my mom was right. I don't know anything about changing human behavior, but now I know people who do. I can recognize that I'm a natural scientist and that we need to bring on people who actually are professionals in changing human behavior. and that we need to engage the community and the community conservation leaders and find ways to fund them because they're really a critical part of this process. Thank you.
Thanks, Barb. That's great, and I certainly resonates with me in terms of humans have created most of the problems, and we can solve hopefully some of them at least through thinking about human behaviour. So we're happy to take questions both online and in the audience here, but maybe just a starting one for me, Barb. So you've worked on vaquitas, part of your career. And obviously the trends aren't great for the species. Based on what you kind of starting to learn about the role of human behaviour and other sciences in driving conservation outcomes, if you had your time again, would you do things differently? And what would that be?
Yeah, thanks for that. I certainly would do things differently. And one of the things that I learned through this, and I should say that the IUCN Conservation Planning Group has really great training in all of these conservation planning aspects I talked about. And when I took that training, I said, well, how do we choose the right stakeholders? And then later I learned about conservation psychologists, and I think choosing the right people to get to the table who are really going to share the vision of recovering the species is a really important step. And if I could have had a conservation psychologist sitting beside me saying, the fisheries department has no interest in making this happen, and they will be in your way the entire time, and you should just move on and try to do something that does not require their cooperation. That would have been very helpful. Would have saved us decades of, sort of beating our heads against the wall. And also the same thing on fishers. There are fishers in the community who are respected, trusted people that could have really helped out the situation if we'd had them on board, and they live in the community, so they'd be there long term. And I think having someone who can understand sort of the entire fishing community and pick the right people to be at the table is really critical.
Great, thanks. Okay, so we've got probably 5 minutes or so for some further questions. I'm watching online. Andy, have we got anything online yet?
Yeah, I've got one question if you haven't got any in the room just yet. This one comes from Jerry, and he said, we're aware that awareness isn't enough, but when there's a clear conflict between livelihoods of species and livelihoods of people and the species persistence, how do we sort of engage with that? And is community engagement and a community champion enough to overcome such a tension?
Yeah, it is always a real problem with, with our situations with bycatch. It's, people putting food on their table. It's food security in lots of areas. And, it is a really difficult problem. And I think there's no easy solutions to it. In the case of vaquitas, there was alternative gear that could be used, and fishers that, if they knew that they could make as good a living doing that, so that's where the economists come in, then, you could socialize that gear and make it work. And I think that's what you have to look at, is you have to look for those shared solutions. And sometimes you're not going to find them, but lots of times I think that there are ways, if we can incentivize the fishers to change their behavior, or whatever group it is.
Great.
Well answered.
We'll take one from the auditorium here. Andy, thanks, Pete. So the question, if I can paraphrase it, Pete, is, we have different types of government across the world, from authoritarian to democracies and all the things in between. And the question is, how can we, what role do those different types of government have, and how can the behavioural science kind of inform how we might work in those different governance arrangements.
Yeah, that again is another really hard question. I mean, I think two of the things that are really important to know in conservation planning is, do you share a vision with whatever the governmental entity is? And if you don't, do you just need to find some other way to work with the community? So I think that's one of the things that's really important. The other thing that conservation doesn't acknowledge very much is understanding how much corruption there is, which is a really, really big problem and is highly correlated with the poverty index in various countries. And I think if you're in a situation where you do not have government support and you do have corruption, I think you just have to find other ways and not think you're going to be able to change those things. And that, I mean, that was the case in Mexico. We were just, there was never any hope. And I'll give one exception for the government, and that is still to be seen. There are international bodies that can lean on governments. So there's CITES, there's various international treaties, and often they don't have much in the way of teeth to do anything, but sometimes even the threat of being able to interrupt international trade is sufficient to motivate at least some action on a part of a reluctant government.
Got another one online here, Barb. Do you know whether taking a One Health or One Welfare approach, demonstrating how human and other animal and plant health and welfare are connected, and promoting solutions that benefit people, other animals and plants, the environment and the planet, leads to human behaviour change in any of these situations? The kind of merits of a One Health approach.
Yeah, the One Health approach, I think, has been one aspect that's been very useful in coastal communities with the dolphins and porpoises that we work with. Getting veterinarians in the field to work with you and to work with the local communities so that they understand that the fish that they're depending on and that the dolphins are depending on are the same fish. And if it's an unhealthy environment for the dolphins, it's also unhealthy for the community. So I think that certainly is a way of reaching out and getting human behavioral change because it benefits their health and their children's health. So yeah, that's a very good point.
Great. We've probably got time for one more. So either online, Andy, if you've got one there that I can't see, or in the room here.
I don't have one.
Okay. Well, thanks again, Barb. And I think everyone, if we can just put another round of applause. I should say for the people, at least at Heidelberg, Barb and Jay will be around after the session. So if there's stuff you want to talk with them specifically, then that offer's available. So thanks again, Barb, for that fantastic talk. So next we're hearing from Dr. Jay Barlow. Jay was also a leading scientist at NOAA in California, as well as adjunct professor at Scripps Institute for Oceanography at the University of California in San Diego. He's currently a marine mammal researcher at the Marine Mammal Research Institute at Oregon State University. He led the development of acoustic technology for monitoring marine mammals and contributed to the discovery of a new species of beaked whale off the coast of Mexico. That's extraordinary. He interprets acoustic data to model the distribution, abundance and trends in marine mammals, which is what he's going to present to us today. So welcome, Jay, and over to you. Thank you.
Thank you all. I'm delighted to be here and given the opportunity to talk to you all. Today I'm going to be talking about listening to beaked whales, what they can tell us about their behaviors, distribution, and abundance. This photograph is of a Cuvier's or goose-beaked whale, Ziphius, and it's really the star of my show. It's got one of the widest distributions of any mammal on the planet, going from ice edge to ice edge. What's the biggest animal you've never heard of? It's likely to be beaked whales. I don't think there probably are many beaked whale experts in this audience. At least some of you may have heard of them. There's the largest Baird's beaked whale found in the North Pacific, 10 meters long and the weight of 2 elephants. But this is a beaked whale, and this is a beaked whale, and these are all beaked whales. Of the things that we call whales, more than half of them are beaked whales. And of the cetaceans, the whales, dolphins, and porpoises, a quarter of them, more than a quarter, are beaked whales. So we have a lot of them. I'm going to talk about mostly that goose-beaked whale today, but also mesoplodons. And most of these little guys are of the genus Mesoplodon. And even though I say little guys, they're still 4 meters long and weigh as much as a horse. So if you take nothing else home from this talk, just remember beaked whales are really amazing. They routinely dive to 1,000 to 3,000 meters depth. That's 3 to 9 Eiffel Towers. How many have climbed the Eiffel Tower? With one breath, up and down 3 times. And that's just typical. They routinely hold their breaths for an hour and can do so for up to 3 hours. And when they surface to breathe, it's only for 2 or 3 minutes, and they go down and do it again. Roadmap, because you're not all beaked whale experts, I'm going to give you a little bit of introduction to beaked whales, their behavior and their acoustics. I'm going to talk about why we use acoustics to study beaked whales. I'm going to talk a little bit about the equipment that I developed when I was at NOAA's Southwest Fisheries Science Center for acoustic monitoring. And I'm going to talk about some mini studies on beaked whale behavior, distribution, and abundance. So here I'm showing 3 days in the life of a tagged Cuvier's beaked whale. And so I have time along the x-axis, that's 3 days, and depth along the y-axis. And you can see that every 3 hours or so, they take these deep dives, and we call those foraging dives because the rest of the time they're not feeding and they're not echolocating. So the blue portions of those deep dives are the times when they're actually actively echolocating and searching for prey. In between, they take these relatively short dives, only 1 Eiffel Tower. And you can see 2 measures of the seafloor depth on this plot, and you can see they're diving down frequently to the seafloor to do their foraging, but not always. They don't have to feed in association with the seafloor. And a lot of them occur in the Bissell Plains where they simply could not practically reach the seafloor. It's 4 kilometers deep. And that little red bar on the far right is the maximum depth of any scuba diver. A colleague of mine, Simone Bauman-Pickering at Scripps, first recognized that all beaked whales appear to have different and species-specific echolocation pulses. Give a couple of 8 different examples here. Baird's beaked whale on the left top has a frequency of 26 kilohertz. Cuvier's beaked whale on the right top has a frequency of 37 kilohertz. But the common feature of all the beaked whale echolocation pulses and none of the other cetaceans is that they all have an upsweep to them. Like most bat species have downsweeps, these have upsweeps. And we can use that characteristic to tell that it's a beaked whale, and then we can use interpulse interval and peak frequency and bandwidth and things like that to tell what species they are. I'm going to talk also a little bit about BW43, which initially was a mystery. We didn't know what that was, but we have some ideas now. So there are 2 main methods to study beaked whales. Most of my career was using visual sighting methods. They have to surface to breathe, but they only spend a few minutes at the surface every hour. So that's bad. They're hard to see in rough seas. They're not spectacular. They don't fluke up. They don't splash. Usually they don't breach. We can see them at generally less than 2 kilometers, and it's almost impossible to identify most species at sea. The females and juveniles for all the Mesophodon species look more or less the same. So unless you have an adult male present, you don't even know what species it is. Acoustics has some advantages. They are often silent. They're not echolocating all of the time, as you saw in that previous plot. But they do vocalize almost continuously for those foraging dives for a period of 15 to 40 minutes, once every 3 hours. And they're kind of like metronomes. They're really regular in their patterns, so we don't have a lot of variation. We can acoustically detect them at up to 6 kilometers, although 4 kilometers is a more reasonable value for most species. And really importantly, their acoustic signals are species specific. So unlike visual, we can actually tell what we're detecting. The way we did visual sighting surveys in the Southwest Fisheries Science Center, we get a big white ship, one of these NOAA research ships. We go out there for 3 to 4 months. We search with these 25 power big-eyed binoculars, 2 of them from the flying bridge deck, and we look for marine mammals. But we didn't see very many beaked whales, in part because they're not spending a lot of time at the surface. So in these surveys years from 91 to 2008, we had 6 surveys. We had a total of 57 sightings of Xiphias, that's the goose-beaked whale, and only 31 sightings of all the Mesophodon species combined, and there are 5 different species there. So we are only having an average of 5 to 10 sightings per survey, which is pretty low for estimating abundance. The methods for doing acoustic surveys are 3. We can tow hydrophone arrays, and I've done quite a bit of that in my career. We can use bottom-mounted recording systems, and my colleague Simone has used a lot of those in her career. But I'm mostly going to concentrate on a drifting system that I developed when I was at Southwest Fisheries Science Center. In the drifting system, we have a package, a spar buoy at the top. So I called them drifting acoustic spar buoy recording systems, or DASPers. The spar buoy contained the geolocation device. It's floating at the surface. We have 2 hydrophones down at about 100 meters depth, separated by about 5 to 10 meters each, and autonomous recorders. So we have to pick these up to get the data back. We did a survey in 2016 where we deployed these DASPers at 22 different locations along the US West Coast. The deployments were relatively short compared to our typical survey, only about 20 days on average, but we had almost 900 detections of this one species of beaked whales on the acoustic recorders. So this is many more detections than we had during all of the visual sighting surveys that we've ever had in this area. If beaked whales could talk, what questions would we ask them? Well, I'd certainly like to know how they're making a living, how they're spending their time, where they're spending their time, what the abundance is, and are the populations doing okay? Well, they can't really talk, but we can listen. And by listening to the echolocation pulses, we can study their diving and foraging behavior, determine species distributions, estimate their abundance, and determine whether their abundance is increasing or decreasing. I'm going to skip over the behavior part because I don't have time in this talk, but we can determine species distributions because each sound is unique. You can see in the stars here the distributions of detections that we had of Cuvier's beaked whale on the left, Xiphias. And you can see that They have a really broad distribution in our study area, but they were really rare in northern cold water. So Northern California to Oregon and Washington, they were very low densities in those areas and had the highest densities. Can you see my arrow? Yep, down here south in the Southern California Bight. We discovered on these surveys a new beaked whale signal type. We could tell because of the upsweep again that it was a beaked whale. It had a peak at, a notch, a V-shaped notch in its frequency distribution at 39 kilohertz. So we called it 39V. We actually went out on an expedition in off of Oregon, led by Oregon State University, and we found a Hubb's beaked whale. We biopsied it from the vessel, we got recordings, and we confirmed that this BW39V is made by Hubb's beaked whale. Now, whenever we hear that anywhere in the world, we know what it is. BW43 was another mystery beaked whale. We thought it was maybe Perrin's beaked whale. In 2018, we had another drifter experiment. We found this great cluster of detections in this one area off Baja California, Mexico. So we organized an expedition with the help of Sea Shepherd. We went down there. We found a very interesting whale. We couldn't tell what it was, but it wasn't what we were looking for because of we could tell from the tooth placement of the adult male. And then finally, just this last summer in June, we went down there again and we got that all-important biopsy, and we found out that it's ginkgo tooth beaked whale. So this BW43 signal that has this upsweep here at 43 kilohertz peak frequency is made by Mesoplodon ginkgodens. So we're still making incremental progress, but we've reached the point where we can actually now map the distribution of most of the northern hemisphere species just from the sounds that we receive. We're still working on the southern hemisphere. Sort of the holy grail of acoustic studies on marine mammals is to estimate their abundance. And so we've been making some progress on that using what are called acoustic point transect sampling methods. This is a distance sampling, so like line transects, distance matters. We have our DASPER recording here at the middle of a circle. This is the maximum detection range. And some of the animals within that range will detect, and some we will miss. And we have this concept called the effective detection radius, which is effectively how far can we detect things. So the number of animals that we detect outside that range is equal to the number that we miss inside that range. And then we can use that radius to estimate the effective study area, the effective area surveyed, and infer some probability of detection as a function of horizontal range. This is the key to getting density, is actually estimating how far you can detect animals. We published a paper on this in 2021, acoustic estimates of Cuvier's beaked whale abundance. And I'm going to go a little bit through the methods that were used in that paper. As I said, we had this 2016 survey with drifting DASPERs. We had the acoustic detections, and about 900 out of 111,000 2-minute sampling periods, our little snapshot, had beaked whales in them. So about 1% of our samples had beaked whales detected in them. The snapshot-based point transect density estimation formula is shown here. So the density of animals per square kilometer is equal to the percentage of snapshots, again, 2-minute sampling periods that had detections, times the group size, and divided by the area effectively surveyed. Remember, we got that area from the radius effectively surveyed, and the probability of detection. The probability of detection is needed because they're not echolocating all the time. So we estimate this effective area surveyed, pi times the radius squared, and we can then estimate abundance within a defined study area as this mean density times the study area. Normally in distance sampling methods like point transect methods, you take an observed distribution of detection distances and infer a detection function directly, and then you have then you can estimate this effective detection radius. We couldn't directly measure the distribution of horizontal distances, but we could measure the distribution of detection angles. So the hydrophones were up here near the surface at 100 meters depth. The whales are down here at, let's say, 1,000 meters depth. And from the time difference of arrival of their echolocation signals at the two hydrophones that are separated by 10 meters, we can estimate this angle alpha. And if we knew the depth that they were at, we could estimate detection range. And so what we can measure is this distribution of detection angles. So the novelty here is that we didn't use an actual distribution of detection distances because we didn't know that, but we used two different distributions: the distribution of depths at which they echolocate and the distribution of these observed detection angles. And from these two, using a method that we call simulated maximum likelihood, we can actually come up with a detection function and that all-important effective detection radius. The probability that they're available to be heard is basically what fraction of time they're actually producing sounds and we're able to receive it. The sounds that they produce are very directional, and so even if they're within a detection radius where we could conceivably hear them, if they're facing the other direction, we won't hear them. But empirically, we measured the average encounter was about 17 minutes long. We knew that the dive cycle duration was a little over 3 hours, and so the fraction of time that they're echolocating is that 17 minutes divided by 191 minutes, or about 9% of the time they're available for us to detect them. We ran the numbers, and from that we estimated these new estimates for that 2016 survey in red, which about 5,500 beaked whales in this 1 million square kilometer study area. And for comparisons, these are the visual survey estimates that have been corrected for all the factors that we can correct for availability and things like that. And we're in the same ballpark. But if you look at the coefficients of variation expressed by the error bars here, you can see that because we have so many more detections, the acoustics give us much more, more precise estimates of abundance. So what we'd like to do with that information is to estimate, are they increasing or decreasing? And now we get to the conservation part. The reason why all of this is important is that beaked whales can strand and die when exposed to Navy sonar and seismic survey vessel sounds, the air guns and other associated sounds. So we have a major conservation issue. We don't know whether the few animals that we've seen on the beach associated with this is just the tip of the iceberg, or whether that's, all that there is. I'm sure that it's not all, the animals we can count on the beach is certainly not all that there is. But we really don't know, is this causing population level impacts or not? So what's going to be very important going forward is having a tool that allows us to estimate abundance precisely enough that we can estimate trends in abundance, so we can look for population level impacts. And I don't think we'll ever convince anybody based on the animals on the beach that we've got a problem, but having a time series of abundance estimates will really help nail this down. And there's a survey going on right now, and they're out there deploying the DASPERs and doing hopefully another abundance estimate soon. I want to thank all of my my funders and folks that have helped me on these surveys through the years. Thank you very much.
Thanks, Jay. That's fascinating, and congratulations on the work that you've contributed to and led. We've got time for about 8 minutes, 7 or 8 minutes for questions. Maybe one for me to begin with. So you've got the use of acoustics to estimate densities and abundance. Is there any other type of information which might be non-acoustic that you think could help you with, kind of greater accuracy and being able to kind of assess the trends?
Yeah, I kind of glossed over this point, but there are 3 important parameters that in this study I did not take from acoustics. So the group size I took from the visual sighting surveys, because we can't yet estimate group size from acoustics alone. The distribution of depths that I used to infer that detection function, that came from tagging studies. And the duration of a dive cycle, that 190-minute parameter, that I also took from a tagging study. But since then, I've been working on methods to estimate at least, well, all 3 of those parameters just from acoustic data. So I can talk about that in more detail, but my ultimate goal is to apply this to animals that we can't tag and for which we don't have good group size estimates from visual sighting surveys, because only about 2 species have really been tagged out of the 24 species, and we need information on all of them.
Great, thank you. All right, well, we might open up to questions. We've got one here in the audience, Lindy. If you. So the question for those online is, how variable are the calls within an individual species?
They seem to be very, very stereotyped, and that's fortunate because we can recognize one of these Ziphius, even if it's coming from the Mediterranean. It still looks the same to us. But what sometimes changes is if you get a very weak signal that's off axis, that's not the focus beam that they're projecting ahead of them, it can look different. And we have a category that I think is really important to have: unidentified beaked whales. There'll be some signals that are just too faint to classify as a species, and you shouldn't be arrogant about it. You should just admit that, I can tell it's a beaked whale, but I can't tell what species it is.
Well, do things change through time? Is that the... Yep.
Yeah.
Yeah, the detection bit.
Yeah, the detection distance could well vary with oceanographic conditions, but mostly the variability that you get in propagation of sound comes when you're in the surface layer, and very small variations in the depth of the thermocline can greatly affect things. But they're echolocating mostly in the homogeneous waters that are below the thermocline down to 2,000 meters. And because those waters are relatively homogeneous, the sound propagation is relatively uniform. So we don't think, I've done the calculations of sound propagation in the coldest and warmest areas, and it doesn't predict that the detection distance would change.
So what do we know about beaked whales around Australia?
Australia, like the United States, it spans a lot of habitats, and relatively little is known about the beaked whales here. There's been intensive studies done in New Zealand because it seems to be a real hot spot for beaked whale strandings. But I would guess that you'd have some of the same beaked whales here: the Gray's beaked whale, Shepherd's beaked whale. You'd probably be getting the northern bottlenose whale, the southern bottlenose whale, and Tasmacetus. I know that you have those in the northern waters, more tropical waters.
Well, thank you all. We've run out of time for today, but thanks to Barb and Jay for 2 fantastic and informative and groundbreaking presentations and the research that you've done. So I think if we can put our hands together again. Okay, so this is our last seminar for the year. So thanks to everyone who have contributed. Thanks to all the presenters and also to all the people who've continued to attend these, both online and in person. I'd like to thank Andy and Tess in particular for all their hard work behind the scenes. Dealing with all the tech has been fun, but I'm hoping that everyone has got a lot out of them. I've certainly enjoyed hosting them as well. So we'll kick off again next year. We'll send out some information around the timeframes and the schedule for that. But if in the meantime, if you're new to these seminars, please make sure that you get onto our mailing list, and Andy will drop some links in the chat there that you can use to do that. And I think we're dropping some links into both Barb's and Jay's papers as well, and just a further advert just to sign up for ARI updates as well. So Andy will put all those links into the chat. So thanks everyone again for attending. Thanks for your efforts through the year. Thanks again to Jay and Barb for today's presentations. Fantastic. And for the people here at Heidelberg, I think Jay and Barb are around for a little bit longer and available to have a chat to anyone who wants to follow up on some stuff. Tracy. Great. That's an added incentive. Okay, thanks all. Have a good day, and see you all in the new year for the next seminar. Thank you.